Good afternoon, everyone. I'm Rabia Zara, a PhD scholar from Department of Zoology of Government College University, Faisalabad. I welcome you all in third International Conference on Applied Zoology 2020 on the behalf of Organizing Committee of ICOS 2020. I will be the moderator of third session, which will include the talks from the topics of parasitology, entomology, toxicology, and pathology. Let's begin our session with the first talk of Dr. Aisha Siddiqa from University of Punjab. She will be sharing her talk on defensive potential of aqueous onion extract against tartrazine mediated hepatic and nephric toxicity in mus musculus. Please, Dr. Aisha Siddiqa.
Assalamu alaikum. I am Aisha Siddiqa from Department of Zoology, University of the Punjab, Lahore. For this conference, I am going to present on the topic defensive potential of aqueous onion extract against tartrazine mediated hepatic and nephric toxicity in mice. Tartrazine is a commercial food dye which is extensively used in food products, pharmaceuticals, and cosmetics. The acceptable daily intake for tartrazine is 7.5 mg per kg body weight per day. If it is overly consumed, it may cause hyperactivity, memory loss, and learning dysfunction. The aim of current study was to evaluate the remedial potential of onion against tartrazine instigated in vivo toxicity in male mice. Whole experimental work was conducted in Animal House, Department of Zoology, University of the Punjab, Lahore. The treatment related to animals as well as dissection were carried out by following the instructions provided by National Institute of Health and Bioethics Committee of University of the Punjab, Lahore. For the experimental purpose, I raised colony of mice. After 4 weeks of birth of pups, I weaned them out and took 4 weeks old 30 male mice and divided them into 3 groups. First group was control group, second was dose group in which 0.14 mg of tartrazine was given, while the third group was dose plus antidote in which along with tartrazine 1 ml of onion extract was also given. After the dose administration period of 30 days, dissection was carried out and biochemical, histopathological, and morphometric analysis were carried out. Now coming towards the results, the body weight during the 4 weeks of experimentation period in dose group, the weight of mice decreased significantly in comparison with the control. The weight of liver also decreased significantly in dose group when tartrazine was given. The weight of kidney also decreased significantly in dose group. Now coming towards the hepatic biochemical analysis, alanine transaminase increased significantly in tartrazine treated group, while in other groups it didn't show any significant difference. Aspartate aminotransferase increased significantly in comparison with other groups. Alkaline phosphatase also increased significantly in dose group. Total bilirubin also increased significantly when tartrazine was given to mice, while total protein decreased significantly in both dose plus antidote group, while it decreased remarkably in tartrazine group. Now coming towards the renal biochemical analysis, urea also increased significantly in both dose and dose plus antidote group, while it increased remarkably in tartrazine administered group. Creatinine also increased significantly in dose group. Now coming towards the histopathology, This is the histopathology of liver. This is the control group, this is dose group, and this is dose plus antidote group. In control group, this is the central vein. These are sinusoidal spaces. This is upper cell, hepatocyte, and nucleus. While in this group, it is showing the congestion of central vein. These are the sinusoidal dilations, binucleated cells, and these black arrows are showing pycnosis. While in this group, black arrows are showing pycnosis, while this is showing necrosis. This is the histopathology of kidney sections. This is the control group, which is showing normal histology of kidney. This is glomerulus, Bowman capsule, distal convoluted tubule, and proximal convoluted tubule. This is the dose group in which these black arrowheads are showing glomerulosclerosis. This star is representing necrosis, and these statics are showing tubular degenerations. While in this group, when tartrazine was given along with onion extract, these black arrows are showing mildly congested glomeruli, and this is the Bowman capsule, distal convoluted tubule, and proximal convoluted tubule. From the current study, I concluded that when onion extract was given along with tartrazine, it protected various biochemical alterations and histopathological lesions to a great extent, and this may be due to the activation of antioxidant defense system of the body. These are the references, and thank you for your attention.
Thank you, Dr. Aisha Sadika from the University of Punjab for such a nice presentation. Now moves towards our second talk of this session by Saida Nadia Ahmed from the University of Sargodha. She will be sharing her research work on amelioration by Syzygium cumini, Frigeria ananissa, Morus macrora, and Olea phrygiana fruit extract on testes, seminiferous tubules, and spermatic cells by CCL4 induced histopathologies in mice. Please Saida Nadia Ahmed.
Bismillahirrahmanirrahim. Assalamu alaikum. I say warm welcome to all honorable guests on Third International Conference on Applied Zoology 2020 that is organized by the Applied Zoological Society of Pakistan and Quaid-e-Azam University, Islamabad, Pakistan. First of all, I'll briefly introduce myself. I'm Dr. Sayyida Nadia Ahmed from Department of Zoology, University of Sargodha. And today I'm going to present the research paper whose title is Amelioration by Syzygium cumini, Frigeria ananissa, Morus macrora, and Olea phrygiana fruit extract on testis, seminiferous tubule, and spermatic cell by CCL4 induced histopathologies in mice. Let's start from the brief introduction that CCL4 is used as a solvent, propellant, and dry cleaning agent, as well as in the formation of chlorofluorocarbons and medicine. Its main route to the exposure to human beings are inhalation, ingestion, and dermal contact. It increases the production of reactive oxygen species that cause massive germ cell death and bring changes in sperm structure, and that leads to the male infertility. While different fruit extracts are used to tackle the situation or to ameliorate the toxicity created by CCL4, like Jambal or Syzygium cumini, it's a medicinal plant. It is used because of the presence of precious phytochemical like carbohydrate, amino acid, tannin, saponin, phytosterol, terpenoids, phenol, flavonoids, and anthocyanin. Just like strawberry or Frigeria ananissa contain different specific phytochemical like allergic acid, anthocyanin, flavonoids, and vitamin C. Similarly, olive or Olea ferruginea fruit contain different antioxidants which are more specific like oleuropein, polyphenol flavonoids which enhance sperm concentration and sperm motility as well. Morus macrora is also our chief source of vitamin C and vitamin K, and along with various anti-inflammatory substances and various antioxidants like beta-citosterol, anthocyanin, alba-furon, and thiamine. The purpose of the recent study was to find out the male toxicity in terms of histopathology of testes and histometry of seminiferous tubule and various spermatic cell against CCl4 exposure, and to find out their amelioration upon post-treatment of Cygin, Cumina, Fageria, Nanissa, Morus macrora, and Olea ferruginea fruit pulp extract in mice. Material and method consist of 5 steps. In step 1, animal distribution and dose profile was finalized. In this study, 60 animals that were divided into 6 groups were used. First group was vehicle control, second was CCl4, and last 4 group include different fruit extract and along with CCl4 treatment. In step 2, CCl4 and fruit extract were prepared. In this study, zero point 0.1 volume of 0.2 ml per kg CCl4 solution was used, and in step 3, dissections were carried out on day 7, and organ and testes were processed, and blood was collected that was sent to UMDC Sargodha for further analysis and lipid profile. In step 4, organ were processed, sectioned, and histologically studies were done. And in last step, histometry or micrometry was performed along with data analysis. It is a little bit more detail of the dose profile. It was the 7-day study on day 0. Vehicle control group were treated with zero point 0.1 ml cornile, while other 5 groups were treated with the same treatment like zero point 0.1 ml of 0.2 ml per kg CCl4 solution was given to all animals. From day 1 to day 6, vehicle control and CCl4 group gain the same treatment of mineral water, while last 4 groups include respective fruit pulp extract treatment that was provided via gavages, and on day 7 the section was performed. Histopathological results clearly show in A section, that is vehicle control group section, the seminiferous tubules were rounded in shape that contain proper rails of spermatogonia, spermatocyte, primary and secondary spermatocyte, and centrally the lumen that was filled with this developing spermatozoa. Around different seminiferous tubule, healthy interstitial tissue was placed with a large number of Leydig cells. In section B, in CCl4 group, you can see here the shape of seminiferous tubule is distorted, as well as there are many hollow spaces in the center, and there is a clump of different spermatogenic cells, and there are no spermatozoa, just only the head of spermatozoa, a tail of spermatozoa are present, and there is also obliteration of interstitial tissue and less number of Leydig cells. In C section, we show CCl4 Cygin Cumini group. The seminiferous tubule are little bit repairing is found in seminiferous tubule, like there are different spermatogenic cells that are arranged themselves in proper orientation, and there is a partial rehabilitation of interstitial tissue. In B section, we show CCl4 Morus macrora group. There are seminiferous tubules with the large and lavish number of spermatogonia, and in the lumen, you can see here the large number of spermatozoa and developing spermatozoa that cover the lumen of the seminiferous tubule, and around each seminiferous tubule, a little bit rehabilitative interstitial tissue along with Leydig cells are found. In E section, we show some CCl4 and Olea ferruginea group. Excellent rehabilitation was seen here. The size of seminiferous tubule was almost equal to the vehicle control group seminiferous tubule, and maximum Leydig cells along with healthy interstitial tissue was present here, as well as lumen was also filled with the developing spermatozoa. as well as proper arrangement of all spermatogenic cell was present here. In F section that shows CCL4 fragilia and ANESA group, the size of seminiferous tubule and diameter of seminiferous tubule was very larger, as well as central lumen was occupied with developing spermatozoa. Here the size and the shape of seminiferous tubule was also polygonal, and there was a little bit rehabilitation of interstitial tissue. Table 1 shows different micrometric parameters or histometric parameter that was calculated, like mean number of seminiferous tubule were maximum in the vehicle control group and less in fragilia and ANESA group. In mean number of spermatogonial wells were greater in Oleopharygenia group and less in Morus necrora group as compared to vehicle control group. Mean number of spermatocytes were greater in CCL4 group and less in Syzygium cumeni group as compared to the CC vehicle control group. Percent nucleocytoplasmic index of spermatogonia was greater in Syzygium cumeni and less in Morus necrora group as compared to vehicle control group. Table 2 shows biochemical results of lipid profile, and here the biochemical parameters were mean plasma level of triglyceride that was greater in CCL4 and much recover are best addressed in fragilia and ANESA group as compared to vehicle control. Plasma level of cholesterol was greater in CCL4 group and reduced or much recovered in fragilia and ANESA group again as compared to vehicle control group. Plasma level of HDL was greater in CCL4 group and best addressed in fragilia and ANESA group. Plasma level of LDL or low density lipids were greater in Oleopharygenia group and much addressed in fragilia and ANESA group as compared to vehicle control group, while VLDL level was greater in CCL4 group and much best addressed in fragilia and ANESA group as compared to vehicle control. Our conclusion is that CCL4 has strong gonotoxic effect, while the post-treatment of different fruit extract like fragilia and ANESA, Syzygium cumeni, Morus necrora, and Oleopharygenia fruit pulp extract lead to the improvement of andropathological signs, while out of 4 used medicinal plant, the results of CCL4 Oleopharygenia and CCL4 fragilia and ANESA were excellent. It shows excellent rehabilitative capabilities. Thank you for your attention and precious time. Allah Hafiz.
Thank you, Saida Nadia Ahmed from the University of Sargodha for presenting such an amazing research work. Now it's turn for the third talk of this session by Dr. Azhar Rafiq from Government College, University, Faisalabad. She will be presenting her talk on determination of prevalence and risk factors of infection with Babesia in small ruminants in Punjab, Pakistan. Please, Dr. Azhar Rafiq.
Bismillahirrahmanirrahim. Assalamu alaikum. I am Dr. Azhar Rafiq, Assistant Professor, Department of Zoology, Government College, University, Faisalabad. The topic of my presentation is determination of prevalence and risk factors of infection with Babesia in small ruminants in Punjab, Pakistan. First of all, the introduction. Babesia are the protozoan transmitted by ticks, also called epicomplexan parasite infecting erythrocytes. Some species are zoonotic and affect human health. Clinical disease caused by Babesia species infection is characterized by fever, hemoglobinuria, and acute anemia. Also involves nervous system like in coordination, abnormal salivation, teeth grinding, and lethargy. Subsequently, death can also occur. Babesia is more abundant in small ruminants, sheep, and goat. Pakistan is the 4th largest milk producing country with the fastest growing dairy sector and from last several years as compared to other countries. The 3 species of genus Babesia, namely Babesia motasi and Babesia carasa and Babesia ovis, are responsible for disease in small ruminants. Babesia motasi and Babesia carasa are considered less pathogenic and infection is most subclinical, while Babesia ovis is extremely pathogenic and instigates severity of the infection with 30 to 50 mortality rate in sheep and goat. goats. The current studies plan to detect Babesia ovis in naturally infected sheep and goat herds of Faisalabad, Doba, Taksim, and Jang district of Punjab, Pakistan. Materials and methods. A total of 892 blood samples in both seasons, summer and winter, from ovin were collected, and the number of sheep 544 and number of goats 348, with a history of high fever or presence of ticks on the body residing in Faisalabad, Dopa, Singh and Jang district in Punjab, Pakistan. Samples were taken in tube with anticoagulant, and the blood is mixed with the 1 ml 90% ethanol stored at 4 degrees Celsius for DNA extraction, PCR, nested PCR, blood smear, and hematological parameters. These are the primer sets used for Babesia ovis DNA detection. And these are the primers used in nested PCR. We also determine the hematological parameters, for example, PEX or cell volume, RBCs, hemoglobin concentration, mean corpuscular volume, etc. This figure then the results. This figure shows the photograph of sample showing positive result for Babesia ovis. This is the PCR product base pair 549 base pair. They show positive Babesia ovis. The M lane is DNA later 100 BP base pair. One is positive control, set 2 is negative control, and sample 3, 4, 5, 6, and 7 are for positive for Babesia ovis. This photograph of sample showing positive results for Babesia ovis by nested PCR. Lane M is the DNA ladder. One is for positive control, the second lane is for negative control, and sample 3, 4, 5, and 6 are positive for Babesia ovis. So we show the at 186 base pair. This table shows the prevalence of babesiosis in small ruminants in summer and winter season in 3 districts, Faisalabad, Toba, Taksim, and Jang. The overall prevalence in summer based on microscopic PCR and nested PCR was in sheep and goat or small ruminants, while in winter the prevalence is low as compared to summer. The prevalence of Babesia ovis in district Jang was higher as compared to other 2 districts. This figure graph show the risk factors. The risk factors are the age, sex, feeding system, housing, and herd, body condition, and dog association. And the statistical analysis showed that tick burden, weak body condition, closed housing, and association with dogs increase the prevalence of the disease. In conclusion, babesiosis were much prevalent in our sheep and goat herds that may lead to huge economic losses in terms of poor growth rate, poor quality wool, and increased treatment cost after disease outbreak. Smear method was not accurate for detection of carrier animal. PCR and nested PCR for the detection of Babesia ovis was more specific and sensitive. Babesiosis sheep and goats were with significant decrease in HB concentration, PCV, RBC, etc. It was observed that herds consist of more than 20 animals. Poor hygienic conditions associated with dogs and having ticks on their bodies were the major risk factor for the spread of babesiosis in small ruminants. Thank you for your attention.
Thank you, Dr. Azhar Rafiq from the Government College University, Faisalabad, for presenting your talk. Now it's turn for our next talk by Aqsa Arshad from the University of Punjab. She will be sharing her talk on reprotoxicity and abnormal steroidogenesis following oral exposure of tartrazine in mice and remedial potential of Allium cepa. Please, Aksa Arshad.
Assalamu alaikum. My name is Aksa Arshad, student of MS Zoology from Department of Zoology, University of the Punjab, Lahore. The topic of my presentation is reprotoxicity and abnormal steroidogenesis following oral exposure of tartrazine in mice and remedial potential of Allium cepa. The brief introduction of this dye is, it is a synthetic food color, imparts yellow color in various food items and belong to the class of azo dye compounds. It's a higher consumption in various dietary items such as biscuits, sauces, noodles, and sweet meat were reported. However, this dye is nowadays highly consumed by children in junk foods and is responsible for various hypersensitivity problems and behavioral changes in children. In addition, noxious consequences are also reported on reproductive system in mice as well as steroidogenesis in both mice and animal models. The objectives of my study were, as this dye is nowadays consumed above their ADI levels, especially by the children and adults. So there is still ambiguity that exists regarding its reproductive toxicity. So present study is executed to explore the reproductive toxicity of this dye, to explore abnormal hormonal functions in mice model, as well as the use of aqueous onion extract as a protective source against the steroidogenesis and reproductive toxicity caused by this dye. Moving towards the materials and methodology, both genders, male and female mice, were bred and maintained in the Animal House of Zoology department, and their required doses were prepared as reported by the literature, that is 0.140 milligram of tartrazine and 0.1 ml of aqueous onion extract. The animal study design was designed in such a way that both male and female mice weighing 14 plus minus 1 gram were selected, 30 male mice and 30 female mice, and both male and female mice were then divided into 3 groups as control group, which was administered along with water. Second one is dose group, which were given 0.140 milligram of tartrazine, and the third one is dose plus antidote group, which were given 0.140 milligram of tartrazine along with 0.1 ml of aqueous onion extract. Both genders were given 30 days of treatment period, and after that they were sacrificed according to the protocol provided by Ethical Committee of Punjab University, and the following parameters were assessed, such as body weight, gonadal weight, biochemical analysis. In addition, histopathological defects of testes and ovarian tissues were also examined. Then next is results of all above parameters. First of all is body weight of male mice, which you can see in this graph that it is decreased, significantly decreased in dose group as compared to the control. The next one is the body weight of female mice, which also showed a significant decline in the dose group as compared to the control. However, you can see in this graph that when dose is administered along with the antidote, which is the aqueous onion extract, the female body weights were resumed to some extent. The next one is organ weight of male mice, which is testis weight, which is also decreased significantly in the dose group as compared to the control. Organ weight of female mice were also showed a remarkable significant decrease in the dose group as compared to the control. But if we compare the organ weights of both male and female mice, you can see that testis weight were decreased in dose group and ovarian weight were also decreased in dose group in the case of both male and female mice. But you can see in this graph that the organ weight of female mice, that is ovary weight, showed a more remarkable or a significant decrease as compared to the organ weight of male mice, which shows that this dye poses more noxious or consequential effects on the female reproductive system as compared to the male reproductive system. Then moving towards the male reproductive hormone analysis, which is the testosterone, which is the main reproductive hormone in males. It shows a significant decrease in dose group as compared to the control in terms of mean plus minus standard error mean. Then the semen analysis of male reproductive system were examined, which includes the sperm count in terms of millions per ml normal sperm number and abnormal sperm number. The sperm count in the male mice were decreased in the dose group as they are treated with the tartrazine dye. Next, the normal sperm numbers were also decreased in dose group. When, however, you can see that when dose group was given, mice were treated with dose plus antidote group, their normal sperm number were resumed up to some extent and was somehow near to the control level. In the case of abnormal sperm number, the abnormal sperm number were significantly or remarkably increased in the case of dose group when compared with the control. So male reproductive hormonal analysis, testosterone and its semen analysis, sperm count and abnormal sperms were significantly increased in dose groups when compared with the control. Then moving towards the female reproductive hormonal analysis, follicle stimulating hormone level were decreased in 4 weeks old mice as shown by the different superscripts. Luteinizing hormone in female mice when treated with the dose group, that is tartrazine, also showed a significant decline as compared to the control. However, when female mice were given dose along with the antidote, means dose plus antidote group, the luteinizing hormonal level was almost similar to or near to the control level, which means that allium cepa has some sort of antioxidant capacity in resuming the level of luteinizing hormone in females. Then moving towards the estradiol level, it was also showed a decline in the dose group when compared with the control. Then moving towards the histology, first of all, histology of testes. As you can see in these slides, the picture A is of control group, picture B of dose group, and picture C is of the dose plus antidote group. As you can see in control group slide, the normal organization pattern of all the reproductive cells of males, that is lumen, properly assembled lumen, spermatozoa, Leydig cells, and the spermatogonia are can be seen in the control group of male mice, which shows the normal organization of all these cells. While in dose groups, you can see that it shows a reduction in the number of spermatozoa. You can see that in the seminiferous tubules, the number of spermatozoa are declined. On the other hand, it also shows the dilation of the seminiferous tubules, and interstitial species also increases. In the case of dose plus antidote group, it shows the some restoration of lumen with the sperm number shown by the red bar, which somehow increase, means lumen is again organized and the sperm number increased to the some extent. However, spermatogonia and spermatocyte cells can also be seen in dose plus antidote group. However, in the case of Leydig cells, they still show the degeneration or reduction in their number, as represented by this blue arrow. And these testis slides were observed at 10x using the H&E stain. Then moving towards the histology of ovaries, both A picture is of control group, B picture is of the dose group, and C picture is of the dose plus antidote group. As you can see in the control group, there is a normal development of oocytes, secondary follicles, antral follicles, properly organized corpus luteum and follicular cells can easily be seen. However, in the case of dose group, you can see that follicle atresia is occurred, as shown by the yellow star. Also, the vacuolations were seen in the case of dose group in the female ovarian structure. Then moving towards the dose plus antidote group, which is a C picture, it shows that increase in the number of follicular cells, especially the primary follicle number is increased, secondary follicle number is increased, as shown by the yellow and red triangles. which shows that somehow the female follicular cells and ovarian structure was resumed when dose was given along with the antidote group. So the conclusion of my above work was that tartrazine has damaging effects on reproductive parameters and also induced some testicular and ovarian tissue damages. Also showed a decline in the levels of male and female hormonal levels such as testosterone level, follicle stimulating hormone level, estradiol level, and luteinizing hormonal level. However, we can see in all these slides that when those were administered along with the antidote group, somehow all above parameters such as body weights, organ weights, and hormonal defects were resumed to some extent, which means that allium cepa, which is aqueous onion extract, was somehow beneficial or show its potential in resuming all these factors to its normal values or near to the control values. So, however, keeping in view all above toxicological problems resulted by this dye, it would be better that this azo dye would be replaced by some coloring compounds that contains the natural ingredients. Thank you.
Thank you, Aksar Arshad from the University of Punjab for delivering such an informative talk. Let's move towards our next talk of this session by Dr. Madiha Arshad from University of Education. She will be presenting her talk on cardiovascular and hepatotoxicity induced by nickel and therapeutic potential of Punica granatum in male Swiss albino mice. Please, Dr. Madiha Arshad.
Assalamu alaikum to third international conference on applied zoology. Here I am Dr. Madiha Arshad from University of Education Faisalabad campus. Today the topic of my presentation is cardiovascular and hepatotoxicity induced by nickel and therapeutic potential of Punica granatum in male Swiss albino mice. First of all, what is nickel? Nickel is a transition metal that is highly resistant to corrosion and it has two oxidation states and it is mostly having different oxidation state used for electroplating to make alloys. Its physical properties are silvery white heavy metals, exhibit shine or glow, good conductor of heat, easily magnetized, hard and ductile. It is usually present in the earth crust, air, atmosphere, with drinking water as pollutant due to the biological weathering, and it can cause serious hazardous effects. In this study, we have checked the hazardous effects of the nickel on cardiovascular system and hepatosomatic systems. And alternatively, we find the ways to reduce the effects caused by nickel. For checking the ameliorating effect of pomegranate was used. Pomegranate as already as you know that it decreases oxidative stress, effective against cancer, diabetes, Alzheimer's disease, anti-inflammatory activity, reduce fatty liver in obese people, and it has 2 to 3 times larger antioxidant abilities. As nickel damages the DNA by producing reactive oxygen species, means it causes oxidative stress that is being reduced by the pomegranate extract, that can be reduced by the pomegranate extract. Materials used in this materials and method animals used in this study were male Swiss albino mice that were placed in steel cages of 8 into 14 inches. They were provided with the standard light and dark conditions, and the humidity was maintained at 40 to 45 degrees centigrade. Chick feed number 14 was fed to the mouse. Water was given in free access ad libitum. Nickel chloride was used as a source of nickel. Its LD50, according to Siam 2008, was 16 to 130, and sublethal doses of this LD50 was managed to provide it to the mouse. Experimental groups were divided into 3 control groups: dose group and antidote group. Control group was not given any treatment. Dose group was given 8 mg per kg of the nickel chloride. Dose 2, medium dose, were given 15 mg per kg, and dose 3 were given 30 mg per kg. Similarly, antidote group contains a single application of antidote uh, pomegranate as 500 mg per kg, and antidote plus dose 1 500 mg pomegranate extract plus 8 mg per kg as nickel chloride. AD plus dose 2 group was given 500 pomegranate extract plus 15 mg per kg nickel chloride. Similarly, antidote plus dose 3 groups were given with 500 plus 30 mg per kg nickel chloride. Now here we come to the results. If we discuss about the lipid profile, because we were checking the cardiovascular effects of the nickel and the ameliorative effect of the pomegranate. So lipid profile was checked. Here you can see total cholesterol level, high density lipoprotein, low density lipoproteins, triglycerides, and very low lipoprotein were increased significantly in dose 1, dose 2, and dose 3, which were given only nickel chloride. That means they cause the arteriosclerosis and hypertension within the blood vessels. And when they were given with pomegranate extract along with the dose, they showed almost normal activities of these total cholesterol, high ATL, LDL, and VLDL, almost normal to the control values. If we talk about the liver functioning test, total bilirubin, ALT, and ALP were also increased. That means the liver functioning was disturbed by the nickel chloride when given separately, but when it was given along with the pomegranate extract, it shows normal values of total bilirubin, ALT, and ALP. If we see the histopathology of heart, first one figure shows the normal heart histopathology, and the other 3 are taken from the dose groups, which shows uh, congestion in the blood vascular system. Similarly, they also shows the ICDD inter uh, likewise fibrosis were also observed from the heart. If we come about the talk about histopathology of the liver, it first one figure shows the normal histology of liver, that is hepatocytes and central blood vessels. Sinusoidal spaces are clearly visible and normal. But in those group, those mice which were given with nickel, they showed necrosis, fibrosis, congestion, or apoptosis. Binucleated cells were more present in the dose groups, whereas the antidote plus dose group shows almost normal histopathology, but minor changes were observed. Thank you so much for your attention.
Thank you, Dr. Madiha Arshad from the University of Education for sharing such an informative research. Let's move towards our next talk by Dr. Priya Latif from Bahauddin Zikriya University. She will be presenting on DNA damage caused by chronic exposure of cadmium in Ketla Ketla. Please, Dr. Priya Latif.
I'm Dr. Priya Latif, Assistant Professor of Zoology at Institute of Plant and Applied Biology, Bahauddin Zikriya University, Multan. Yeah, I am presenting my abstract entitled DNA damage caused by chronic exposure of cadmium in the Ketla Ketla. As you all know, tremendous increase in population, urbanization, and industrialization has led to the deterioration of aquatic environment due to the addition of untreated sewage, industrial or domestic waste directly into the environment, aquatic environment. The phenomenon of aquatic pollution is not new, but the pace of industrialization has exhibited its negative impact on the aquatic environment. These pollutants include metallic ions, polycyclic hydrocarbons, dioxins, polychlorinated biphenyls, and other xenobiotics in the natural waters. Among these resources, heavy metals are essential for normal physiological processes, but in very small quantity. While their excessive concentration may cause hazardous effect for aquatic organisms, such as they may cause behavioral, biochemical, and histological changes, and ultimately reduce growth and mortality of the fish. As you can see in this image, the stress factors including metal and metalloids may cause different physiological and histological changes in the fish, and they may cause imbalance between oxidants and antioxidants by the production of reactive oxygen species, and also they may cause oxidative stress that lead to lipid peroxidation, DNA oxidation, protein oxidation, that ultimately lead to impaired fish health or even death. Therefore, during this project, I have observed and calculated dose and time-dependent DNA damage caused by cadmium exposure to the fish, that is Catla. Okay, the methodology that I have used, first of all, the 120 days old fish fingerlings were procured from fish hatchery, and they were acclimatized to the laboratory conditions in the cemented tanks 15 days prior to the trials. Determination, first of all, then acute toxicity of cadmium was observed or determined, sorry, in terms of 96 LNC50 and lethal concentration to the fish Keta. And after this determination, I have selected 4 different sublethal concentrations. Those are 2/3, 1/3, 1/4, and 1/5 of 96 LNC50 concentrations. Then fish were exposed to these sublethal concentrations and sampling was done after each fortnight is 14 days, 28, 42, 56, 70 and 84 days. And after each fortnight the sampling was done, and total there was 6 fortnights. Then comparison of cadmium exposed fish was done to positive control intraperitoneally injected with cyclophosphamide and negative control, that is unstressed group. And DNA damage was determined by using comet assay technique in the peripheral erythrocytes of Ketla. This whole procedure for the comet assay was followed for the preparation of slides to determine DNA damage and to observe them with fluorescence microscope to take images that was further processed by using comet scored software to take images and measure DNA damage. Okay, the DNA damage was estimated in terms of 3 different parameters. First one is the mean percentage of damaged nuclei that was calculated by adding type 2, 3 and type 4 DNA damage. while GTI or genetic damage index was measured or calculated by using the described formula, and comet length of damaged cells were measured by using the Critic Comet Score software, and the cumulative tail length of comets was calculated by adding the comet tail length of all the examined cells. And this was also after taking the images from the Fluorescence microscope. Okay, in the results, first parameter that was determined to detect DNA damage was damage nuclei. As you all can see in the upper graph, during the 6 fortnight, cadmium will cause significant damage, and it was variable. Maximum damage was observed during the fourth fortnight, and these damage nuclei were observed during first fortnight, while the damage nuclei order was fourth fortnight, followed by second, fifth, third, sixth, and the least damage was observed during the first fortnight. Now coming towards the sum of damage nuclei by treatments, least damage was observed in negative control, which was not exposed to any metal, and the maximum damage was observed by positive control, the fish that was intraperitoneally injected with cyclophosphamide. Then the damage nuclei among the treatments was observed in two-thirds, one-third, followed by one-fourth and one-fifth. Moving towards the genetic damage index, GTI was also significantly variable during all fortnights and among the treatments. Least damage was observed in negative control, while positive control followed by two-thirds, one-third, one-fourth, and one-fifth. That was treatment dependent. As the dose of cadmium decreases, the GTI was also decreased in terms of DNA damage. Moving towards the genetic damage index that was observed during fortnights, cadmium will cause significantly higher DNA damage to the nuclei as GDI values were significantly higher after fourth fortnight, while it was lower after first fortnight. And however, after 28 and 70 days of exposure, the damage in the nuclei become non-significantly different at p less than 0.05. In terms of accommodative tail length, this damage was observed during first fortnight, while it was maximal in the fourth fortnight. And in terms of treatment, so this damage was observed in negative control, while it was maximum in two-third control, followed by positive control, one-third, one-fourth, and one-fifth. So, in the conclusion, cadmium will cause significant DNA damage to the fish erythrocytes, which was dose and time dependent. That is all about my topic. Thank you for your attention.
Thank you, Dr. Frihalati from Hardin Secure University for presenting such an amazing research. Let's move towards our next talk of this session by Shabnur Iqbal from Government College University, Faisalabad. She will be sharing her research work on synergistic effect of Moringa oleifera leaf and cinnamon cassia bark methanolic extracts against bisphenol A-induced hepatotoxicity in rats. Please, Shabnur Iqbal.
I'm going to present in Third International Conference on Applied Zoology. I'm going to present synergistic effects of Moringa oleifera leaf and cinnamon cassia bark methanolic extracts against bisphenol A-induced hepatotoxicity in rats. Bisphenol A is an organic synthetic compound composed of two phenolic groups, and its very high production volume chemical widely used in different plastic industries, and its major environment pollutant, and it's very harmful, and it's cause different harmful effects in cells and tissues, and its main mechanism is oxidative stress, and it produces different toxic free radicals, and its exposure might cause different diseases like cardiovascular disease, brain development, obesity, hypertension, thyroid dysfunction, etc. in human, terrestrial, and aquatic animals. These are the sources of bisphenol A. We use because bisphenol A is very toxic, so it's need to be mitigated. So we used medicinal plants, And we use two medicinal plant, Moringa oleifera and Cinnamon acacia, and because due to presence of different nutritive components in Moringa oleifera that are vitamin C, beta carotene, protein, phenolphthaleins, and ascorbic acid. While cinnamon acacia also have different effective biological activities like cancer, insecticidal, antioxidant, anti-mutagenic activities, and it also have cholesterol lowering effects, antimicrobial effect, and anti-inflammatory effect. Hypothesis: the co-administration of cinnamon acacia bark and Moringa oleifera leaf methanolic extracts cause synergistic effects to mitigate bisphenol A-induced hepatotoxicity. Aims and objectives: investigation of individual and combined cumulative effects of cinnamon acacia bark and Moringa oleifera leaf methanolic extracts against bisphenol A-induced hepatotoxicity through various bioassays like hematologic analysis, biomarker of oxidative stress, liver function, and histologic analysis in Sprague-Dawley rats. We use two plant extracts as we already discussed, cinnamon acacia and Moringa oleifera, and we procured from local market of Faisalabad and identified by Department of Botany, Government College University, Faisalabad. And we selected the healthy Sprague-Dawley rats and approved from the ethical committee, and then we kept in ventilated cages under the light condition in animal house of GC University Faisalabad. We acclimatized the rats for 7 days and then selected similar weight rats and randomly divided into 6 groups, and each group contained 5 rats. Negative control only fed water and commercial feed, while vehicle control administered with olive oil via oral gauge, because we make the bisphenol A dose in the olive oil. That's why we use the olive oil as a vehicle control. Positive control group is labeled as BG1, that only received bisphenol A at the sublethal dose of 30 mg per kg per body weight uh, on the basis of already reported LC50. Other treated groups are assigned as CBG2 that received bisphenol A, and cinnamon acacia MBG3 received bisphenol A and Moringa oleifera, while CMBG4 received cinnamon acacia, Moringa oleifera plus bisphenol A. And the effective doses of cinnamon acacia and Moringa oleifera were selected from previously reported as LD50, and they were treated for 59 days, and the treatments were 3 days in a week. These are the extraction preparation method. We selected 4 parameters for the analysis of different parameters: hematologic analysis, liver function test, oxidative stress, histology. These are the results of hematologic analysis that show that the bisphenol A cause toxic effect, while the BMCG4, that was a co-administrative group of cinnamon acacia and Moringa oleifera, that shows synergistic effect as compared to its individual doses. And in the hematologic analysis show that each results of platelets, WBCs, and other white blood cells like lymphocytes, granulocytes, monocytes, they show same result, that BMCG4 show the synergistic effect in due to the combined effect of the Moringa oleifera and cinnamon acacia as compared to the individual doses. And these are the results of hemoglobin and RBCs along with their indices. They also show the positive synergistic effect in after the co-administration of cinnamon acacia and Moringa oleifera, while the bisphenol A treated group show significant toxic effects. These are the results of liver function test. They are also showed that synergistic effects of cinnamon acacia and Bengalophila in BCMG4. The oxidative stress biomarker also show the same results that BCMG4 showed synergistic effects as compared to their individual doses. These are the result of histological analysis that show the clear toxic effects of bisphenol A, uh, that are the congestion, hypertrophy, pycnosis, and necrosis. These are the results of plant extract treated groups, and they also show the synergistic effects after the co-administration of cinnamon acacia and moringa oleifera as compared to their individual doses. Conclusion: bisphenol reduce the RBCs and hemoglobin, while it significantly increase the platelets and WBCs, and also increase the liver enzymes, as well as cause severe histological alteration like congestion, pyknosis, necrosis in liver portal and central vein areas. It's also reduce significant oxidative stress and reduced antioxidant like catalase and glutathione, while the Moringa leaf and cinnamon acacia bark extract show synergistic immunity effects and improve different parameters like hematologic analysis, biochemical analysis, histopathologies, and oxidative stress as compared to their individual doses. Significance of study: use those products which are BPA free, ban the BPA and similar products, while cinnamon leaf and cinnamon acacia bark extract co-administration have synergistic effect at the effective dosage and might use to make phytomedicine to lower the liver-related disorder. Moringa leaves and cinnamon acacia bark extracts might also cause use as a supplement at their effective doses in combination to reduce the risk of liver diseases. Thank you for your attention.
Thank you, Shatnur Iqbal from Government College University, Faisalabad, for presenting such an important research work. Now it's time for our next talk by Yusra Fiaz from Jinnah University for Women, Karachi. She will be presenting her talk on comparative study of insecticidal activities by formulated fish emulsified biofertilizer on growth and productivity of Solanum lycopersicum tomato. Please, Yusra Fiaz.
Assalamu alaikum to my all respected seniors and dear fellows. My name is Yusra Faiz and I am a graduated student from the Department of Zoology of Jinnah University for Women, Karachi. Today I am going to represent my research work in the form of presentation which entitles as the comparative study of insecticidal activities by the formulated fish emulsified biofertilizer on the growth and productivity of Solanum lycopersicum. The major aim of this research is to prepare an organic biofertilizer by the utilization of dead and waste fishes, which creates a big mess and pollution on land. This pollution is usually generated by the fishing sector and fishing industry. The second other major reason is the excess use of chemical fertilizers, DDTs, pesticides, and insecticides that slowly degraded the fertility of soil. Moving towards our next slide, that is the abstract. In this research process, three different types of dung were used to evaluate and compare with our research product, that is fish emulsified biofertilizer. Number one is organic fertilizer. Number two is cow dung. Cow dung is mainly converted into two different types of treated pods, that is chemical fertilizer and controlled pods. In this whole research process, it was observed that the fish emulsified biofertilizer has a great resistivity against the pest infestation, and it gives better and best growth of the plant and the food quality. The other is comparative and treated pods and their replicates are very susceptible to the insect infestation, whereas their germination and growth rate is very low. Introduction. There were many authors that described the disadvantages of chemical fertilizers and the advantages of biofertilizers. One of them is Khan. He proposed in their research that the latest use of chemical fertilizers slowly degraded the quality of the soil in the agricultural fields and leaves it unable for the further growth of the crops because the soil is highly toxic and acidic. Too much use of chemical fertilizers in the agricultural fields also has an adverse effect on the surrounding environment and on human health. When it goes into the water bodies, it contaminated the water bodies. In the results, the loss of biodiversity due to the oxygen depletion. The other is Dow and Kim. He stated in their research that the consumption of fishes increasing day by day. So to overcome with the waste and dead fishes and remnants of large fishes, latest technologies were introduced. A certain type of bacteria were inoculated into this trash that converts the trash into the lipid-rich fertilizer that aids and helps in the growth and development of the plant. Preparation of fish emulsified biofertilizer. So the fishes were weighed before and after drying. The process of drying done in the oven binder to remove excess water. Then these all fishes were dumped into a container and covered with a green shade to prevent any damage from the animals. After a month, when the shade is removed, we saw that there were water in the fishes and they were not completely decomposed. So to overcome this problem, we add some organic fertilizer in it, and after a few weeks, it gives best and useful results in the form of decomposed fishes. So this decomposed fishes fertilizer is placed on a newspaper for air drying. After this, this fertilizer is ready to use. We crush some of the larger balls. We can see that the germination that gives in the fertilizer that the germination given by the fish emulsified biofertilizer is very high, and their fruiting capability is the quality of the fruit is also very good, and hardly insect infestation seen on the stem of mealybugs. But if we talk about the insect infestation of other replicates, other pots, treated plants, so we can see here that the control pot is totally destroyed by the infestation of mealybugs. It also has orthoptinian infestation seen on their leaves. The second one is the chemical fertilizer. The chemical fertilizer pot leaves has the mealybugs infestation on and under the leaves. Same goes as with the organic fertilizer and ends in the destruction of the whole plant. We can clearly see in the chart that the infestation is high in the early and the end stages of the plant. But if we look at the fish fertilizer replicates, it has a very low amount of infestation, as it has a capability to resist infestation of insect and pests. Discussion. Khalid. He stated in a research that the biofertilizers involves only in the build-up of the chemical of the physical properties of the soil by enhancing its biological and chemical properties. But in the recent research, we focused on the formation of the biofertilizer by the use of dead and waste fishes to combat with the waste pollution, managing pest population, and also enhancing the productivity of the crop. The last but not the least, Vakas, he stated in a research that the byproducts of fishes were used as a nutrient supplement to to enhance its plant cell activities by enhancing its biological and chemical processes to speed up the plant growth. But in the recent research, it was observed that it is a priceless thing. Fish fertilizer is a priceless thing, that it controls pest infestation without any hazardous effect on the crops. It gives us good productivity of the plant and the crops. Our future perspective is what? Now, it reduces the pollution. If we use waste fishes and converted them and recycled them into the fertilizer, it gives us best fertilizer, and also it reduces the pollution. It also reduces the uses of DDTs, chemical pesticides, and insecticides, as they have the ability to resist insect pest infestation. It also has essential nutrients like NPK, nitrogen, phosphorus, and potassium. Plus, it has a calcium in it, so it speeds up the cell activities. If the selectivity speeds up, it has greater amount of growth, production of the crops and productivity. Thank you.
Thank you, Isra Fiaz from Jinnah University for Women, Karachi, for delivering such an amazing talk. Let's move towards our second last talk of this session by Nayab Khattak from Kohat University of Science and Technology. She will be presenting her research work on prevalence and hematological effects of Haemonchus contortus in Khajlo sheep Kohat. Please, Nayab Khattak.
Bismillahirrahmanirrahim. First of all, I would like to say thank you to ICAS 2020 to give me opportunity to present my research topic on third international conference on applied zoology. I am Nayab Khattak, Department of Zoology of Kohat University of Science and Technology. The research title of my topic is Prevalence and Hematological Effect of Haemonchus contortus in Galjo sheep cohort. Introduction of my research topic is Haemonchus contortus. Haemonchus contortus, commonly known as red stomach worm, the wire worm, or the barber pole worm. Adult worms are found in the abosom of goat and sheep. It is estimated that an infected sheep with 5,000 Haemonchus contortus worms may lose 250 ml blood per day. Pathogenicity. The pathogenicity of Haemonchus infection is mainly related to blood loss. It is cylindrical and has a striking reddish appearance due to its blood-feeding habitat. Distribution along worldwide. Haemonchus contortus is distributed worldwide. Haemonchus contortus cause hematological and biochemical disturbances and produce an adverse effect in sheep and goats by drop of the normal value, especially in erythrocytes, lymphocytes, hemoglobin, packed cell volume. Diagnosis. Haemonchus can be diagnosed based upon the following characteristics. Number 1, clinical signs of anemia. Number 2, low packed cell volume. Number 3, pale mucous membranes dehydration. Number 4, weakness, retard growth. Number 4, the edema are the common clinical sign of haemonchus. Objective. The objective of my research topic is number 1, to find out the prevalence of Haemonchus contortus in Galjo sheep at cohort. Number 2, to evaluate the impact of Haemonchus on the hematology of Galjo sheep. These are the objective of my research topic. I collect stool sample from sheep in district cohort. These are the microscopy pictures of my sample which I collected from sheep, and you guys will see the Haemonchus clearly in these two pictures. These are the microscopic picture, my research topic. Baseline characteristics of the study population. I collect sample from infected sheep and I check it gender, area, and age in month wise. In area, I collect sample from Ustarzai, Forest Hill, Kachapaha, Center VRI. I collect overall 203 sample. In gender wise, male is number is 86 and female is 170. Total is 203, and these are female are mostly infected. In age in month wise, I collect sample through 6 to 12 month, which is 31, 12 to 24 is 56, 24 to 36 55, 36 to 48 is 28, and 48 to 60 is 34. Oral sample is 203, and these are the percentage negative sample, positive sample, which are shown in these pictures. I collect blood sample from infected, healthy, and normal sheep. And indicator which I use in this is MCV, MCH, MCHV, PLT, RBC, HB, and HCT. And difference in which I notice in these are normal, healthy, and effective sheep. The difference is +6, and indicator which is used is MCV. Infected, healthy, normal, which is difference is -1, and an indicator which is used is MCH, MCHV. The difference is +8. PLT indicator which is used in the difference is +3. RBC indicator is used, which is the difference is -1. HB indicator is used in normal, healthy, and infected sheep, which is -5. And HCT indicator which is used, the difference is noticed in -8. Conclusion. Conclusion is that it is concluded from the current study that female sheep were mostly infected, which I shown in previous pictures, with the Haemonchus contortus in girl sheep and district cohort. Further concluded that majority of infected sheep showing severe anemic health recommendation. Detailed epidemiological study of Haemonchus contortus infection we carried in girl sheep and district cohort. Molecular and therapeutic measure is recommended to be performed. Awareness campaign may be arranged in the study area about Haemonchus contortus complication and control measure. Thank you for your attention to give me opportunity to be to present my research topic. Thank you so much. Stay safe.
Thank you, Nayab Khattak from Kohat University of Science and Technology for presenting such an astounding talk. Now it's time for the last talk of this session by Dr. Hira Altaf from University of Agriculture, Faisalabad. She will be presenting her talk on blood parasites of on dogs and insight into potential diagnostic methods to identify the threat. Please, Dr. Hira Altaf.
Assalamu alaikum. I am Dr. Hiraltaf from University of Agriculture, Faisalabad and Department of Parasitology. The topic of my presentation is blood parasites of on dog, an insight into potential diagnostic methods to identify the threat. As you all know, dogs are one of the most prevalent and popular pet animal in the world. In addition to keep them as a pet, they are also used for many practical and recreational purposes due to special characteristics such as agility, strength, and sharp senses. They are selected and trained for many services such as hunters, guardians, detectors, and rescue dogs. Dogs' population are highly prone to many parasitic infections, particularly with those of blood parasites. Among parasites, blood parasites lead to numerous deadly diseases in dogs. In all parasitic diseases, hemoprotozoans cause most lethal infections as they flourish in blood and the major reason of erythrocyte destruction. Blood parasites, for example, Babesia, Theileria, Trypanosoma, Hepatozoon canis, and Dirofilaria, are major cause of infections in dogs. Hemoprotozoans cause different effects on dogs, such as anemia due to destruction of RBCs, anorexia, pyrexia, pale mucous membrane, staggering gait, and corneal opacity. Yes, if we talking about the global prevalence, globally 40.1% to 57.3% of the population is declared positive for different blood parasite. Here is an insight of the worldwide prevalence of Babesia species. For example, in India, it caused 26.67% prevalence. In Japan, it's 8.8, China 9.3, and Nigeria 1%. If we move towards the next, which is hepatozone canis, canis are highly prevalent in the whole world. In India is 30%, Turkey 22.3%, in Italy is 57.8%. If we look at the trypanosoma, in Italy is 50%, Portugal 41.1%, and in Spain it's 62.5%. But it's less in USA as 0.6%. Uh, Hemoprotozoans are also very prevalent in Pakistan. For example, in various studies shows Babesia prevalence in Lahore and Narwal as 42%. In If we look at the hepatozone, it's 5.8. In Lahore and Narwal, in Islamabad, is 22.40%. In Multan, it's 8%. Control of blood parasites. These hemoprotozoans are transmitted by different arthropod vectors. It's a need of time to control of blood parasites. It's a challenge that requires measures focused on arthropod factors and environment based on the epidemiology of these parasites in the given area, because parasites depend upon vector for its life. Ticks are responsible for the transmission of many important hemoprotozoans such as Babesia, Theileria, and hepatozone. Another widely prevalent hematoprotozoan such as trypanosoma is transmitted by Thabanus and stomach species of biting flies, while Dirofilaria is transmitted by mosquitoes. Proper identification is necessary to control their transmission by vector control. So it's a need of the time to utilize different molecular and serological based methods to identify infectious organisms which are not unable to diagnosed by classical methods. There are various diagnostic methods are used for the diagnosis of blood parasite. For example, conventional diagnosis on the basis, first of all, the clinical signs, then physical examination of dog, and then move if we move towards a specific identification, then we can use microscopy with a combination of thick thin smear and staining. Microscopy is the most common and cheap method is in the microscope examination. uh, Accuracy is limited, but it's considered a feasible and the simplest method. But in microscopy, we should take care, because it might be influenced by stain debris and cells in different ways. In addition, blood smear preparation techniques also has a role in proper detection of parasites. Microscopy is a good method. It can provide the information regarding the presence of parasite, but it cannot provide any information about the genotype of parasites. Eh, if we move towards the next methods, which is ELISA, CFT, and agglutination, serological methods are a very good tool in with combination of other methods to detect parasites. For example, ELISA test in combination of thrombospondin related adhesive protein is better than ELISA test alone for the identification of babesiosis. Uh, Although ELISA is a valuable and reliable tool for the detection of parasites, but it also has some limitations. So, in the recent years, molecular methods are necessary to diagnose uh, parasites. For example, PCR, loop mediated amplification, reverse line blot hybridization, and quantitative fluorescence resonance energy. Polymerase chain reaction (PCR) gained a lot of importance as a proven method due to its accuracy and sensitivity for detection of parasites. Various species of parasites can be distinguished through PCR, which could not be distinguished morphologically through the blood smear methods. So, information about strain and subspecies may be increased through a combination of genetic sequence analysis and PCR. Uh, if we prefer a method, then PCR the best method to identify the microorganisms, protozoans, which present in very small amount, which is limiting factor for light microscopy, and PCR is much sensitive in this respect. Um, the combination of methods is also a good procedure to identify and diagnose these infections. Thank you.
Thank you, Dr. Hira Altaf, for such an amazing talk. I hope you all have enjoyed this session with such an astounding talks. Now it's time for our fourth session, which will be moderated by Mr. Saddam Hussain, and it will include talks from the fields of natural products, cancer biology, epidemiology, and recombinant DNA technology. Stay tuned with us.